Hope you guys all had a great week and great session last week with Lauren. Me and Heather were in Alabama last week, so I hope it all went really well. Today we'll talk about, well, we have a special guest, Hannah Blackburn. She is with the Texas Apiary Inspection Service, and she'll be talking about Tropi. It's a mite that's found in Asia that could be potentially devastating in the United States if it ever got here to the States. Followed by that, we'll have Heather, who will talk about products of the hive. So, Hannah, I'll let you go.
Okay, so my name is Hannah Blackburn. I am with the Texas Apiary Inspection Service. Um, we are the state regulatory agency for beekeeping as well as honeybee health inspectors. Just in case you didn't know who we were. And today I will be discussing the Tropicalaps mite, Tropicalaps mercedii specifically. Um, so if you need it, there are a number of QR codes throughout the presentation that will take you to a number of different resources. So some are our office and some are others related to the presentation. So feel free to snag those at your convenience. And I guess we'll go ahead and get started. So my presentation will discuss some of Thailand's beekeeping related to the Tropilaps mite. We'll also discuss the Tropilaps mite and some of their biology, distribution, classification, and things like that, as well as some of the current monitoring and management techniques that are known. But something that you'll hear me say throughout this presentation is there's still a lot we don't know. So even though we do have some idea of some monitoring or management techniques that may have been successful for a project, there is still a lot more that we need to learn about this mite. So take all of that with a grain of salt because the more people research this mite, the more we'll know and the better we'll be able to combat it. Then I'm going to do a brief overview of how Varroa and Tropilaps mites are similar or dissimilar, and then throw some extra resources and tell you how to report it if you think you found it. So in Thailand, um, there are about 5 species native to Thailand. So the Tropilaps mite is from the Southeast Asia area. Of the 11 species in the world, Thailand has those 5 native species and 1 introduced species, which is Apis mellifera. So they do manage a number of colonies of Apis cerana and Apis mellifera. And these are some of the mites. These pictures are some of the bees that are present in Thailand. So as far as Thailand beekeeping goes, there is a beekeeping industry. They have a number of colonies, about 480,000 colonies, as well as 31,000 beekeepers. Most of the bees that they keep are very calm and docile. They don't wear very much protective gear as far as what we would consider wearing protective gear in the US. Their climate is relatively tropical with wet and dry seasons, hot and humid temperatures. With seasonal management, they will have single deeps. They do restrict the entrance because they are concerned with some of those pesky hornets that have been spreading around the US. They deal with those on a regular basis. So they do restrict the entrances of their colonies to decrease issues with hornets. And then they keep very little brood. And because of that, they do limit swarming as well. As far as mite management goes, they do treat monthly for the Tropilaps mite and the Varroa mite. So this is an example of an apiary in Thailand. These photos were taken by Kim Skirm, which is the Massachusetts apiary inspector, I believe chief apiary inspector. I will mention it a little later, but in 2024, a group of beekeepers, researchers, and apiary inspectors went out to Thailand. And so this photo was taken on that trip, as well as the next photo. So in this next photo, you can see how minimal the protective gear that they wear while working their hives is. In some images, you will see people wearing a pith helmet, but in general, most of the time you'll see beekeepers just not wearing anything, just about. Um, and that is related to how docile their bees are. So, as far as the beekeeping, honeybee industry in Thailand, these are some of the products that they will distribute and sell. And with this slide, it shows some of the fiscal income from those products. So I don't have a slide that compares like US products versus Thailand products, but you can see that this is about a 38 million US dollar industry in Thailand. So for them, their biggest challenges are finding forage for their bees, as well as dealing with the Tropicalaps mites. Another graphic that I was given is that Thailand beekeeping calendar. So you can see that same image at the top where the beekeeper is not wearing very much protective gear. But if you notice, they treat very regularly and they have different practices very regularly. So October, November, December, January, February through April, May, June, and July through September. That's the entire year right there. They do have a few flows. So they'll have the wildflower flow, the lychee and longan flow. And then there are a number of treatments that they use. They do usually incorporate 1 to 2 brew breaks. They'll treat with 6 different mite treatments and they'll adjust those throughout the year. Um, then they do have 2 big honey flows where they pull honey weekly. And then compare that to the US northeastern US beekeeping calendar. So 1 to 2 brew breaks, that sounds familiar. But with the US beekeeping calendar, they'll have 3 colony buildup periods where the colonies just grow and grow and grow. There are about 3 mite treatments, depending on the beekeeper, that may be applied, and 2 big honey flows. So if you notice that Thailand slide showed something almost every single month going on. Whereas with this northeastern US beekeeping calendar, there's bigger spaces in which these things occur. So, what about AAA lapse mites? So, AAA lapse mites, there are 4 known species of AAA lapse mites. Previously, there were only 3, but I believe it was 1961. AAA lapse mercedesi was distinguished from AAA lapse clarii. Tropicalaps mercedesi and clary are important whenever people talk about Tropicalaps mites. Generally, they're talking about these 2. But for the purpose of this presentation and for the purpose of most of the warnings related to Tropicalaps mites, you're going to be hearing people talk about Tropicalaps mercedesi. Both of these mites did originate in Apis dorsata, so the giant honeybee, but as time has passed, they have found their way to Apis mellifera. This is relevant because Apis mellifera is what we have in the US today. Um, so basically, they have migrated from Apis dorsata to Apis mellifera. Apis dorsata generally have a seasonal migration between November and January, which can help control those Tropi mites that are present. Additionally, they build single combs along a tree line, which on my next slide, I'll have an example of that. Um, Whereas Apis mellifera, they are moved for pollination seasonally, but as far as them building their nests, they have multiple combs within a colony. So in 1961 or thereabouts, it was established that Tropicalaps mercedisi had moved from Apis dorsata, which means they still occupy Apis dorsata colonies, but they were observed in Apis mellifera colonies. One of the observations was an association with field rats, but those rats were associated with honeybee colonies. And so that it was one research paper and it mentioned it briefly. There is, yet again, more research that needs to be done on this topic, but that is something that led to the realization that they were in Apis mellifera. So as far as the Apis dorsata, you can see on the left where these Apis dorsata colonies will just, each of those frames, each of those layers of wax is its own colony. So these are all sister colonies. And it is very common to see Tropicalaps mites wandering between each of those colonies. So they are quite mobile as far as mites go compared to Tropicalaps mites. The Compared to Varroa mites, the Tropicalaps mite is quite a bit more migratory and movement friendly than that of Varroa. So they can be seen going from colony to colony to colony along those tree limbs. That is relevant because of the way we keep beekeeping, the commercial beekeeping today. Even hobbyist beekeepers have their colonies somewhat relatively close to each other. So it would not be uncommon or not visible for the Tropicalaps mites to move from colony to colony to colony within a yard if they were to arrive. Now, as far as mite dispersal goes, there is some record of it happening during drift or robbing or foraging or potentially even mating, but most of the dispersal is believed to have occurred by human means. So, moving frames of comb from one colony to another, rotating boxes, moving colonies for pollination purposes, or even selling bees to other individuals. This map shows the current distribution, the current known distribution of Tropicalaps mercedisi in its association with Apis mellifera. So they originated out in the Thailand area, Southeast Asia area, but with more recent records, it has been found that they have made their way to Georgia and Russia, which is a good little ways from Thailand. Not US Russia, but I mean, not US Georgia, but um, they have made their way to Georgia and Russia, and that report was distributed in 2024. Um, so this map is starting to grow. The longer time passes, the more red you're noticing on this map. With that, you can see the spread of the Varroa destructor mite. So they started over on the right, and they were in 1952, 1950s, they were in Japan, China, and Russia. But by the 1980s, they had made their way to the US and Canada. So even though the Tropilaps mites have not been dispersing as quickly as Varroa mites did, we are keeping an eye on this pest because we do not want Tropicalaps in the US. Nobody wants Tropicalaps in their area, to be honest, because of how devastating they can be to colonies. So, with that in mind, a group of beekeepers, bee researchers, and apiary inspectors went to Thailand in 2024, funded by Project Apisim. If you are not aware of Project Apisim, look them up. They are a great resource. And I will have a link to one of their YouTube channels later in my presentation. But in early 2024, a group of apiary inspectors, beekeepers, and bee researchers went to Thailand to learn about Tropicalaps mites. A lot of the slides and information that I will be presenting came from that trip. So their intent was to go learn about Tropicalaps mites, get some of these apiary inspectors to know what they look like so that they could come back and inform other apiary inspectors and get us on watch. Um, if we don't know about something, we can't be prepared for it. We can't look for it. So the following November, so November 2024, they hosted a Tropicalaps workshop in Auburn, Alabama. And I was one of the apiary inspectors that was allowed to attend. We discussed what Tropicalaps was. We watched a lot of videos. We had some hands-on with dead Tropicalaps mites, not live ones. No one is bringing live Tropi into the US. But we had some hands-on so we could look at the debris samples and do a few other things. And then we also discussed about what are we going to do if Tropicalaps makes it to the US? So this next slide, there is a Tropicalaps mite wandering along the frame. So if you look, um, it's it's just I have a few videos throughout my presentation and I added these so that y'all could see what they look like. You can see how quickly this mite moves across that frame. There's very fast movement. It kind of stops and starts, but it's rather quick. So this next slide has a honeybee with a Tropicalaps mite arrowed in red and a Varroa mite arrowed in blue. And you can kind of see the size difference, but I will have a better example of the size difference later in my slide set. As far as the life cycle goes of Tropicalaps mites, they reproduce much faster than Varroa mites. So this graphic is from the National Bee Unit in the UK, and I thought it was a great resource, so I got their permission to share it with y'all. Maggie Gill is a researcher for them, and she does a wonderful job. Um, so this shows you the difference in how many mites are produced, either in a worker cell or a drone cell, over that period. So basically, most cells have one mite, one female mite, but can host several. Tropicalaps and Varroa can coincide and share a cell together. And you'll see in a later video of some of the reactions to them sharing a cell. As far as Tropicalaps mites goes, they will enter the cell in the third, fourth, or fifth instar, so between days 6 and 10. But they will oviposit after the second day in the cell, and they'll lay a new egg every 24 hours. Um, Basically, they will produce about 2 to 3 mites from a worker brood cell. So in this graphic, it shows 3 mites for Tropicalaps versus 1.6 mites for Varroa in a worker cell, and 4.5 mites in a drone cell versus the 2.6 in a drone cell for Varroa. This next slide, it's more for your information than something that you'll have to look for. These images are taken with a microscope. So if you don't have a microscope, it's not the end of the world. But I just wanted to show you some of the life stages of the Tropicalaps mites. So those dudonymphs in adults, those last three at the top, will be the ones to emerge from the cell. Um, once the workers emerge and or the drones emerge. Um, then at the bottom, you can see the difference in the sexes of Tropilaelaps mites. So this is getting into the nitty gritty, but I thought it was cool and I like sharing it with beekeepers whenever I discuss this. You can see how that plate along the center of their body doesn't fully connect for the males versus the females where it connects fully. So on this next slide, we have some debris sample. And you can see that Tropicalaps are circled in red. So two of those are really, really light colored. Those are dudonymphs. But the rest of them kind of show up a little bit better. You can see that the ones that are upside down, you can see the plate down the center of their body, as was shown in the last slide. That is one very helpful way of noticing them on a debris sample. I had the opportunity to look at some debris samples, and with all of the wax and this and that and the other, you have to have pretty good eyes to notice them on a bottom board debris sample. And then we also have that varroa mite circled in blue. As far as biology and behavior, with Tropicalaps mites, they are not known to be on the bees for very long. So they are known to spend anywhere from a few hours to a few days versus the period on the bee, the phoretic or dispersal phase of Varroa, where it's 3 to 13 days, which can be an average of 7 days. So if you compare 7 days to approximately 1 day, that's a big difference in the period of time it takes them to go from emerging from a cell to being able to go and start reproducing again. They can be transferred from adult bees or seen running along frames during this period of time. At this point, the best we can tell is that they lack the morphological adaptations, the mouth parts, to feed on adult bees. So that's part of the decreased phoretic phase is with the varroa mite, they actually have to have a period on the bee where they feed before they can go and start laying eggs. Well, that's not the case for Tropicalaps. Additionally, one of the benefits or good things that has been researched is how long can they survive without brood? At this point, they need brood to survive. That is what we know. If they are going to go from cell to cell to cell reproducing, they need that brood, those larval and pupal bees for feeding and to lay their eggs and so on and so forth. So, one of the things that was looked into was, well, how long can they survive without brood? Because if they can't survive without brood, then we've problem solved. We just go broodless for a while. But one of the things that was found was as far as surviving on adult bees, they can only survive about 1 to 3 days. Surviving on comb or other hive materials, it's about 5 days, a little bit more than 5 days. Surviving on dead bees or dead pupa would be approximately 7 to 8 days. So this is good news. This is not bad news. This is good news. But then there's that reproductive phase where they have to be under the cell. It's believed that they consume the hemolymph of larvae and pupa, but more research is still needed on that front. They do prefer larvae that are at least 3.5 days old. They use multiple sites to feed. So this is another relevance as to why Tropicalaps are not that great, not so good for bees, is they will have multiple feeding sites. So if you look at the image on the right, you can see those black dots on that larva, and those black dots are each of them feeding sites. With Tropilaps, they feed multiple sites, but with Varroa, they'll open one site and continue feeding from the same site. So you'd never thought you'd hear me say this, but in some cases, Varroa is the nicer cousin of the mites. So with Varroa, like I said, they feed one site. Tropolaelaps, they feed multiple sites. This can lead to deformations in the bee once it emerges as an adult. And then additionally, there is no significant preference that is known between drone or worker brood for Tropolaelaps mites. Then oviposition. So they do not need to feed before they can start laying eggs. This is another bad thing about them. And they prefer larva or prepupa, but will lay on pupa as well. So here are some lookalikes of the Tropolaelaps mite. If you notice on the left-hand image, we have a few different critters here. So we've got our regular old Varroa mite, our Tropolaelaps mite in the bottom center, and then a pollen mite and a Berula fly. So the pollen mite looks more like the Tropolaelaps mite than any of these others to me. But if you notice, it's more circular versus the oval of Tropolaelaps mites. Now, Tropolaelaps mites and Varroa look similar to a certain extent because they have that oval shape. Then on the right, we have male and female Varroa mites, and there has been some comparison as far as appearance of those two Tropolaelaps mites. So we have our size comparison. What does Tropi look like compared to Varroa? Well, this is to scale. We have a Varroa and a Tropi side by side. And then at the bottom, we have the US penny with those two side by side. Then finally, the bad news. If you have trouble reading the 0 on a US penny and the date, you're going to have trouble seeing Tropolaelaps mites. They can fit within that 0 in a US penny. So if you have a US penny next to you, have a look at it and see how small that is. So this next slide shows an image from Project APISM as well as a video from the National Bee Unit. What's relevant in this video is you can see those front two legs moving as if they're antenna. So that's a really cool feature. They use their front two legs for examination of things. So let's go into the visual identification and morphology and things like that. So morphology, the bodies are longer than they are broad. They are about 1/3 the size of a Varroa. So 1.5 millimeters in length and 0.5 millimeters in width. They do have that reddish, light reddish brown color, and there are 8 legs, which can be visible from above when they die or when they're moving. But as I mentioned, those front 2 legs, they use similar to antenna. They have their own antenna, but they will use their front 2 legs similar to antenna for sensory detection and things like that. If you're looking for them on a frame, you're going to look for fast moving objects. So if they're on the brood frame, you can see them on the surface of the comb. They can be found throughout a colony. Another thing that I will show more videos of is they have a stop-start motion where they move, stop, move, stop, move, stop. So that is indicative of the movement of the Tropolaelaps mites. If there are high infestations, you may find them on adult bees or and when they're on the adult bees, they're not commonly found between the abdomen and the thorax. So then if you're looking for damage, the damage is very similar to that of Varroa. which is unfortunate because it would be nice if there was a distinction between the two types of damage. But to date, the research has found the damage is very similar to that of Varroa. You'll notice the spotty brood pattern, chewed down brood, bald brood where the cells have been uncapped close to the emergence of the bee. Something else you may notice is workers dying at emergence with their proboscis extended, so with their mouth parts extended. Um, Additionally, you may notice deformed wings because they do spread deformed wing virus, as well as other deformities based off those multiple feeding sites. And then there may be mite bodies in the bottom board debris. So here you can very clearly see that stop-start motion. They move, they stop. They move, they stop. And you can also see those front two legs being used as antenna. I do have a few videos within my slide set because I find this very interesting. I find this very, the more you see it, the easier it is to call it out. So I wanted to include some of these videos in my presentation. So this next video is from the UK National Bee Unit. So you can see there's the gravid female, the fat female. She is harassing a few of those Tropicolaps mites, a few of those Varroa mites. And it's found that Tropicolaps kind of tend to do that whenever they're sharing a cell with a Varroa mite. And then if you notice the bottom center, that mite is not pregnant, whereas this other mite has that extended body. She is gravid. So as far as Varroa versus Tropi, the movement is slower for Varroa than Tropicolaps. So Tropicolaps move quickly with that stop-start motion. Tropiolaps can be found throughout the hive, so all along the comb, versus with Varroa, they tend to stay near the brood frames. Tropiolaps have a very short phoretic phase, anywhere from an hour to a few days, compared to that 7-day average for Varroa. Tropiolaps at this time, with the research we have to date, are only known to feed on the brood. And it is believed that they feed on the hemolymph of the brood, whereas Varroa are known to feed and survive on adults and brood and focus on the fat body. But as I said, more researches need to be done on a lot of different topics. Tropicolaps can oviposit without immediately feeding, whereas Varroa need to feed before they can oviposit and lay eggs. More research is needed on monitoring, which I will discuss monitoring here shortly. There are genetic markers for miticide resistance with Tropicolaps mites. So we do know that Varroa can and have developed resistance to some miticides. But of that research, it wasn't done on Tropi. But Tropi, we have found genetic markers for miticide resistance. So that is something that could potentially come down the pipeline. Tropicolaps are known to vector many viruses and possibly others that we're not aware of. And lots of the viruses are very similar to that transmitted by Varroa. Colonies collapse much faster, and I do have a slide later in my presentation that kind of gives you a timeline. Um, they double their population in about 7 to 15 days versus that of Varroa, which take about 30 days. And they reproduce 2 to 3 times faster than Varroa mites do. So here we have another video. And like I said, I'm going to have a few videos here, but some of these videos show, oh, here's a good one. You can see where they've got that chewed down brood, some melted brood, and those are some of the indications that you could have a Tropicolaps mite infestation. So that's a good representation of those. Here we have a mite on a queen, a mite next to pupa, And you can see how it blends in so well with that wax. So as far as monitoring goes, these are some of the techniques that have been used to check monitoring efficacy. So with Tropi Labs monitoring, they did visual inspections on adults, visual inspections on brood comb, and visual inspection for bald brood. In Thailand, usually they said if they see bald brood, they know it's probably time to treat, that they probably have Tropilaps mites in their colonies. Additionally, one of the research, or a few of the research actually, have done uncapping of the brood. Now, this is a very great way of checking for Tropilaps mites. You look actually in the cell. The problem with that is it does take so long to conduct the uncapping. Like usually you're taking capped brood and you're uncapping about like 100 cells or more. And that takes time. And then even once you get it uncapped, you have to be able to see the mite. With the alcohol wash or powdered sugar shake, because of the decreased phoretic phase of Tropicalaps mites, that's not as accurate a method for monitoring, but it was looked into. Then there's the sticky board, the checking the bottom board debris. Oh, excuse me. And then other things that were checked were the bump test, where you take a frame with capped and uncapped brood, you shake the bees off, and then you take that frame and you bang it. So if you notice the tray on the bottom right, that's an example of the debris left over from a bump test. Basically, you take that frame, you bang it multiple times on both sides, to try to dislodge any mites or anything that you could then look at under a microscope and check for mites and things. Finally, they also did eDNA testing, where you would take a swab or a Q-tip of sorts and swab the entrance and some of the cells to see if there's any indication of Tropilaps mite DNA on the frame. So here is another one where that mite just blends right on in with the frame. And then we have this video here. You can see where they've uncapped and like they've got that bald brood look to them. Some of these that are kind of raised up a little bit, but that was just another video. So as far as some of the Tropicalaps mite damage, we have brood reduction and decline of development, which, I mean, we're not surprised there. That spotty, irregular brood pattern, chewed down brood, bees emerging with their proboscis or tongues extended, dead bees, deformed bees, either wings or otherwise, bald brood or uncapped pupa, which you can see a few of those on this image here. Holes in the cappings. So some of those cells, you'll notice that the bees have like opened it up a little bit and didn't bother to close it back up. So that's what that's talking about. There may be some absconding of colonies with Tropilaps mite infestations, as well as lowered honey yields. Additionally, since wax moth and small hive beetles are secondary pests, it is known that wax moth infestations increase. I believe that if we had Tropilaps in the US, especially in Texas, we would notice more small hive beetle infestations. And then this next slide actually shows a comparison of some of the viruses associated with Varroa and Tropilaps. So this is just one set of samples, but in that set of samples, you could see some of the similarity between the viruses that are present in both mites. So this next slide shows it's a medium infestation to my understanding. And you can just see those mites all over the place. You can see that stop-start motion. You can just see them moving around. Um, so as far as those monitoring techniques, so as mentioned earlier, as uncapping pupa was 92% effective, but it's time consuming and hard to see the mites. The powdered sugar shake and alcohol wash were less than 40% effective when checking for mites because of that short phoretic phase. The sticky board, 100% effective. Great for detecting them, but it's time consuming and hard to see the mites. The bump method, which we use in the USDA National Survey and have used for years, did come out to be 73% effective, which is great. But it does kill some of the larvae. Finally, the eDNA method is very effective, but at this point, there's very few labs that offer it, and it's quite costly at this time. So as far as management methods, so we've got some non-chemical methods listed here. But of those, making splits or brew breaks was the best method for Thailand beekeepers. It's their golden standard. So there was some research in the beehive thermal heating pads, but more research is needed before it can be considered a great method. Additionally, we have some of those chemical methods. So formic acid and thymol were both in the Apiary Inspectors of America research, both came up as great methods. But I have been asked a number of times about oxalic acid use. And I did find that in the National Bee Unit, they found that oxalic acid vaporization or sublimation was somewhat effective. It was less than 70%, which isn't great because when you think about the fact that if one mite survives, she can start the infestation all over again. Less than 70% still leaves 30% of your mites. But it was something that was mentioned, something that has been researched, and I knew it was something Texas beekeepers were going to ask about. So in this next slide, we have day one versus day 20, and you can see the decrease in bees. You can see that spotty brood pattern. And these are videos from the National Bee Unit. They were real gracious and shared some of their research, some of their videos with me for my presentation. So we're getting towards the end of it. Are Tropicalaps mites really a problem? And the conclusion that we've come to is yes, they will be. And they are currently in the sense that they're expanding their geographic range. So all it would take is one Tropicalaps mite to make it to begin an infestation because of how fast they reproduce and how quickly they travel. They do reproduce faster than Varroa. They feed exclusively under the wax capping, but they move very quickly. And with their feeding, they do leave bee deformities, scar tissue on those adult bees as after they've emerged. Another problem is that the damage is very similar to that of varroa mite. So even once we got Tropi mites in the US, which it is assumed that they're coming and it's a foregone conclusion, they're not here yet though. But once they were to arrive, detecting that damage would be quite difficult because of how similar it is to varroa damage. They do spread viruses and weaken colonies, and they wipe out a colony if they're left untreated. And that's my next slide. But as far as monitoring techniques, they're very hard to detect, and we don't have a lot of great techniques out there. And management, we do have the splits or brood breaks as far as non-chemical treatment. But as far as chemical treatments, there's not a lot of options out there for us. So those are some of the things. In Thailand, when they were asked, how long will a colony survive if left untreated? They said about 1 month. Just 1 month. So here is a slide with some of the additional resources. That QR code will take you to the Tropi Stop webpage. And they've got some great handouts. They've got this debris sample, like test yourself postcard. And on the back of that, they have the QR code with the answer key, just in case you need it. Additionally, we have this QR code for Project APISM's videos. During that trip to Thailand that they funded, they actually sent out a videographer and recorded a number of great videos, and I highly recommend checking those out. Some of the researchers that I found, followed, and learned from, Jeff Williams and Rogan Tokash were part of that Auburn workshop that I was able to attend. I have heard seminars from Dr. Samuel Ramsey as well as Dr. Jeff Pettis. And then Dan Etheridge and Maggie Gill from the UK's National Bee Unit were very gracious. They've got some videos on YouTube of some of their research. They even recently did another trip to Thailand to do more research. So I see more coming from them in the future. As far as more information is needed, that is absolutely the case. There is so much we do not know about this mite. So let's get to what to do if you think you found it. Report it. Absolutely. Call our office, send us pictures or videos. We Depending on what we see, we can come out and collect sample, either debris sample, wax, a variety of different substrates. And then we can store it and send it off to the USDA. There is the understanding from across US with the USDA is they would prefer that those items are collected by an apiary inspector instead of going outside of the chain of command, basically. So, um, if you suspect it, absolutely report it to our office. We are more like we were receiving the quote unquote murder hornet um, reports, and they all turned out to be cicada killers, but we would much rather know than not know and allow it to spread. So what is Texas doing to stop triple elapse mites? Well, we are working with Apiary Inspectors of America, the Texas A&M AgriLife Extension Service, and other groups and discussing different things. We have had the opportunity of attending that workshop in Auburn in November, as well as the January 2025 Apiary Inspectors of America meeting, and where we discussed planning. What are we going to do if AAA Labs makes it here? We're not trying to sit and wait for it. We're trying to be proactive. Additionally, I am doing public talks to different bee clubs. So if you know of a bee club that is interested, have them shoot me an email. My email will be on the last slide. Um, and I'm happy to line something up to discuss AAA lapse mites to different bee clubs. I've done a few talks already, and I plan on doing quite a few more. Additionally, we are doing the USDA National Honeybee Survey, where we do that bump method to try to detect triple elapse mites. And I'd be happy to discuss that with anybody. So thank you very much for your time. And if there are any questions, I would be happy to address those.
Perfect. Thank you. Fantastic. So are there any questions about AAA Labs?
Yeah, I have one question. Something that was brought up where she was saying what was working, what wasn't, and I noticed that the hygienic or the VSH didn't seem to do as well, if I got that correctly.
Well, there's been a lot of research in whether people are using hygienic or VSH bees in
Mhm.
association with tropicalaps mites. I don't think there's been a lot of research on that front.
It hasn't. Okay. I didn't know if there was something that different between the Aurora and the triple. I mean, I can't.
Tropi.
Yeah. Uh, the difference in the sensitivity of the queen or the colony that can sense it out because something's different about the two.
I don't know if there's been any or much research into like hygienic behavior versus non-hygienic bees. in relation to Tropi bites.
Okay. All right.
So unfortunately, I don't have an answer for you in this
All right.
regard.
Thought I'd ask. Thank you very much.
Sure thing.
Well, because in Thailand, right? They don't really have like a structured breeding program, and I think the USDA in Baton Rouge, I think they're having plans to bring pole line, the VSH like the stock out there to see if like that could be a trade to at least keep the AAA labs at bay. But I don't think there's been like any research at all that I'm aware of.
Yeah, not to my knowledge.
Okay, so Mindy said, is there a time frame that they may get into the United States?
So it only took varroa mites like from the 50s to the 80s to get to the US. So at this point, we've already passed that window compared to varroa. So there's not really a time frame of when they're expected to arrive. No. Um, that would help like determining things, but the hope is that they never make it to the US. The truth of the matter is, as with a lot of different things, it's just a matter of time. We just don't know how long.
Okay, are essential oils effective with Tropi?
Not to my knowledge. I don't believe there's been much research on that topic. I know that thymol was listed as thymol, formic acid, and then there was a little bit of research on, but those aren't like essential oil as essential oils.
Yeah, and I guess you were in Auburn. Which treatments did they try? They tried formic acid. I think they tried like heat. Did they try like any essential oils like Apigard or thymol?
Email me and I can totally tell you because I still have copies of that
Sure.
research. And I believe that research is published on those PAM videos
Is it okay?
for from the Auburn trophy trip. I believe all of that research is published in those PAM videos.
Okay,
But
perfect.
if you email me, that's why I have my slides still up. If you email me, I'm more than happy to try to find the answer for you. Um, but if I don't know the answer, shoot me an email. Or if I don't answer your question to the way you'd like it, shoot me an email and I can tell you it again.
Yeah,
Or if
then
you know somebody who needs us to talk at a club, like have them reach out to me. I'm more than happy.
yeah, and I mean Shay made a good point. The world is much smaller today compared to the 50s, and so that
Oh
I mean that
yeah.
definitely opens a lot more doors, right? Like we have more globalization, and so it's something that we
And
should
that's one
be,
of the things
yeah.
that the National Bean Unit is super concerned about. They've got sentinel apiaries all along their border because they're like, oh, if Georgia and Russia have got it, it's just a matter of time before it gets to us. And they do port trapping and a whole bunch of other stuff. Um, and they get other bee species other than Avis mellifera coming in through ports. Anyways, I'm sidetracking. Yeah,
No,
the
and
National
it's a really
Bean
good
Unit
point.
is super
Yeah.
cool.
Well, and with Varroa, right, when they first came to the United States, like by the time we detected them, beekeepers were already moving bees across the country. And so it'd be a
Oh yeah.
situation where we detect it almost too late, even if we do have the sentinel apiaries or, we do have
And
some kind of port that,
that's one of the problems with if they make it to a commercial operation, then they're going to spread all over the US. Um, so we don't know if they're like which way they might get into the US. There are a number of ways they could make their way here, especially if there's hive debris or dead bees or whatever for them to feed on. Um, if they were to get on a ship or something and make it all the way across. But it's one of those things we would much rather be informed and try to be prepared. Like you can't fully prepare for something you haven't had, you know, but we would like to have ideas. And that's why we're working with the Apiary Inspectors of America and other groups to try to be as planned and prepared as we can be.
Are you guys working with like the Canadian inspectors? Because I know they have like a lot of them buy bees from like Australia, right? And so that could be a
So
logical way they could get into the United States.
correct. So in Canada, we do actually, they're part of the Apiary Inspectors of America group, or there are a number of Canadian apiary inspectors part of that group. Through them, we learned that once Russia announced that they had AAA lapse mites, they completely shut off all purchases from Russia and Georgia, but they had been purchasing from Russia and Georgia. So we are in communication with them. And I mean, I've had conversations with a number of them, and I think that they would let us know if they found it so that we could all be prepared. But it's one of those like they were buying from Russia. It's possible that that could have been a thing. They're buying from other countries. It's possible that those other countries could have it. The map that I showed is just those that have been confirmed by someone who knows what they're looking at, by genetic testing, by this, that, or the other. Um, so even though that map shows a lot of red, there could be more red out there. And the National Bee Unit's map shows a bit more red than our map.
Yeah, so Billy said, will this presentation be available for reference? I think you're gonna post this on the YouTube page, right?
So I know that like these are recorded, right?
Yes. Yep.
Okay. So I know that y'all have the recording for the Master Beekeeper program. I was planning on speaking with Garrett or Heather and trying to get a snippet of my presentation put to give to TBA for their resources. And then like basically because it's recorded, Garrett and Heather can broadcast it however they want. I personally am scared of being on YouTube. But if it makes its way there, it makes its way there.
Okay, perfect. And then Shane said, how do I know the difference between AAA labs or a difference with AAA labs?
So if you're just looking at, because this is the uncapping brood is a VSH trait, um, the difference is visual inspection, seeing that mite, seeing some of the different damage types. But right now, the best way of telling if you have trophy is to see the mite. Either see it moving on the comb, see it on the debris sample, see it different ways. Now, that may mean you need to get a hand lens, like a little magnifying glass that you can look at your debris sample, because they blend in real well with some of that trash that the bees clean off the frames. But there's not a clearly defined difference in the damage from Tropi as that of Varroa.
Okay, perfect. I don't see any other questions. So if there's any other questions, yeah, please leave in the chat. But yeah, Hannah, I really appreciate your presentation. It was really, really fantastic.
Absolutely. And if anybody has additional questions, they are more than welcome to email me, email our office. We are the Texas Apiary Inspection Service. And then my email has been on the screen for a while, so you've had a chance to write it down. Um, but yeah, I'm going to be talking about Tropy for a while now. I had avoided doing presentations, and I can no longer use that excuse.
Yeah, and just one thing about the apiary inspection service. So one reason we have the master beekeeping program, we require beekeepers to register with the apiary inspection service because they're doing such great work across the state. And so even though it's optional, Hannah can attest to that. We do want people to register with the service because they do a lot of really great work. They also provide inspections, they provide they're definitely doing like wasp monitoring across the state. So they do a lot of really great work that's important for our industry. So that's really one reason that we require registration with the TAAS because of all the great work, including what they're doing with AAA labs.
And if you find if you find American foul brood or you suspect American foul brood, you're legally supposed to report that. If we find it and confirm it, if you're registered with us, we can notify you. We won't say, oh, your neighbor has it, but we might say, people in this county, check your colonies more frequently because American foulbrood has been found in your county. Like, we're not going to give you somebody else's info, but we definitely will notify beekeepers if there's something like we get contacted about mosquito spray or like other pesticide sprays. And if we're notified, we can inform beekeepers. So that's another perk of being part of like being registered and being in our system is you can be notified about different things.
Yeah, and I do also want to briefly about how the TIS in Texas differs from other states. So I know you guys are with Texas A&M AgriLife Research, whereas some of the other inspection services are with the state department services. And so do you want to talk about that just a little bit, just so people are aware?
So some states, it just really depends. Some states, most states are supposed to have an apiary inspector of some kind. So some of those are plant inspectors that kind of got added honeybees to their agenda, whereas others are like designated specifically for honeybees. Texas has the designated specifically for honeybees. We are housed under the university. There are other apiary inspection services that are housed under universities, whereas some are housed under the state. Our office was created before the Department of Ag was created, and so we're housed under the university instead of housed under the state. Additionally, some states require that every beekeeper be registered, like mandatory. If you own bees at all, you have to be registered. Texas doesn't do that. Some states, like every state requires, or just about every state requires some sort of movement permit or health certificate or both for migratory movement of bees across state lines. So if you're shipping bees on comb across state lines from or to the state of Texas, you need to have a permit and you need to have a valid health certificate either from the state you left or from us once you get into the state. So we do a lot. Our primary inspections are that of commercial beekeepers. Those migratory beekeepers that are migratory or migratory beekeepers that move across state lines, those are our primary focus in spring and fall. But any beekeeper in the state of Texas can request an apiary inspection. There is a fee associated with that, but we would come out, look at your colonies, answer questions. If you want mite wash or nosema or miticide resistance testing done, we are working with somebody who's doing amitraz resistance testing. Um, those things are possible through our office. And we're a great resource. We try to be at the Texas Beekeepers Association events. We try to be at different events across the state to inform beekeepers and show you examples of some of these pests and diseases. We try to be a resource. That's what we are.
Okay, perfect. And just kind of last note, yeah, Russ had a really great kind of comment. And he basically said, like, hopefully this will never get here, but that is wishful thinking. So it has the potential to be devastating for the industry. Having said that, it will require major changes in management of bees. And I believe that brood breaks are the key. Since mites can't feed on adult bees, a brood break with isolation of the queen in the colony is really the key to this. Yeah. So with Varroa and then even Tropicales mixed to the United States, brood breaks is going to be really important. And so, understanding when your colony begins producing large amounts of brood and trying to target those brood breaks is going to be regardless if you're managing the Varroa mites or whether you're managing Tropicales. And so, one thing I say with Varroa management is, there's certain times of year when your colony has a brood break. So like around late October, early November, into the spring into around February. That's a really great time to apply oxalic acid or one of those treatments because all those mites are outside the brood and are back of the bees, so it'll be easy to manage them that way. You can also add your own brood break to your colony by, caging the queen for a couple weeks. And so you can cage the queen. You can buy these devices at any type of bee supply store, or you can make your own kind of wired mesh to cage a queen in one small area. And by caging her for a couple weeks, you can get that brood break, especially if your colonies are high, and you can treat them with oxalic acid or one of those other treatments. So brood break's always going to be the key. And if you can kind of learn how to target those brood breaks or add a brood break, that's going to be key to not only Varroa, but also Tropi lapse.
Yep.
Okay, perfect. Well, I think we'll take 10 minutes for Heather to get all set up. But yeah, Hannah, I really appreciate it. And I really enjoyed your presentation.
You. Thank you. Yeah, I'm gonna get back. I think I'm checking wasp traps this afternoon. So I'm gonna head that way and get that started. But thank y'all for having me, and hopefully you learned something, and good luck on the exam because I'm sure Garrett's gonna throw some of these questions on there for you.
Thank you, Hannah.
Absolutely.
Thank you.
Thank you.
Okay,
Okay,
Thanks,
perfect.
I'll
Hannah.
talk to y'all later.
We will come back at 10:15, and that'll give us time to get our presentation set up.